Hi everyone and welcome to Miss Estruch Biology, and in this video we're going through all of topic 3. If you do want to skip ahead to a particular part of the topic, then just click the time codes along the bottom. But first I want to say a huge thank you to BioSnip for being this video's sponsor. So BioSnip is a completely free resource for your A-levels. They provide you with a weekly newsletter with relevant biology stories that have been in the news. So if you're struggling to find the content reading around the subject, and it might take you ages to find relevant information, then this newsletter is perfect for you. Once you sign up, it's all automated, and every week you'll be sent a newsletter with the articles that have come out that week in different sources, and they make sure there is a link to the A-level spec, so you could talk about it in the essay or at interviews if you have any university interviews coming up. Due to the applied style of A-level biology, these articles could also really help you to develop your skill set of being able to analyse unknown information, which is so important for the application questions and the critical analysis questions in the A-level papers. These newsletters are just a 5-minute read, so in that short space of time every week you can gain information on a wide range of topics, from immunology to ecosystems to plant vascular systems and cell differentiation. These newsletters are completely free, and the benefits they can provide you are amazing. It takes just 10 seconds to sign up. Either you can use the QR code just here, or I'll put a link to the form in the description as well. In addition to the newsletter, you might have seen me share their Instagram on my Instagram before as well, because I really think that the content that they're posting is so valuable. They have lots of feed posts explaining little bits of the content, but also I love the quizzes that they post in their stories as well. So if you haven't already checked them out on Instagram, definitely go and check out BioSnip on Instagram and start following them for all of that extra free help as well. So once again, thank you BioSnip for sponsoring this video, and definitely go and check out all of their amazing free resources. But for now, let's get into topic 3. So we begin with surface area to volume ratio, and this is where we have this particular formula of whatever the surface area of an organism is, you divide it by its volume. And the relationship between how large an organism is, or it could be the different shapes it has, are linked to the surface area to volume ratio. So this concept has a big role within this topic, where we look at how different cells or even organ systems have adaptations to maximise gas exchange and other types of transports across surfaces. So here we just have an example where we can see this pattern of the larger an organism is. Now in this case we've just got cubes to demonstrate, but the larger an organism is, the surface area divided by the volume, or the surface area to volume ratio, will decrease. So what that means in terms of biology is small organisms, like single-celled organisms like amoeba, have very large surface areas compared to their volume. And that means that they don't have to have any additional special adaptations to meet their needs in terms of transport across their surface. They can just diffuse gases across their surface, and because they've got such a large surface area compared to their volume, they'll be able to get enough gases in and out to meet their metabolic needs. However, larger organisms, because the surface area is so much smaller compared to the volume, that means that if we were just to get gases into the organism through simple diffusion, the cells right in the centre of the organism are so far away that the gases wouldn't be able to reach there in sufficient time for their metabolic needs. Now on top of that, larger organisms typically have higher metabolic needs as well. And what that means is the chemical reactions within a cell are happening faster. So that is another reason why larger organisms have to have adaptations for mass transport or just for transport across cells. And that's what most of this topic is looking at, the adaptations that organisms have for their exchange surfaces to make exchange more efficient. So some of the key adaptations are the villi and the microvilli. So within the small intestines, those two particular types of tissue and then cells to maximise the absorption of digested food. The alveoli and bronchioles, so they're involved in gas exchange. Spiracles and tracheoles involved in gas exchange, but that's for terrestrial insects, whereas alveoli and bronchioles are for mammals. Gill filaments and lamellae is the gas exchange adaptation surface for fish. And then we'll be looking at plants as well. They exchange gases through the stomata on the leaves, but we'll be looking at some of those adaptations as well. Now, in addition to that, when we get to the idea of the mass transport as well, we'll see a link to gas exchange, whereby most of these gas exchange surfaces have a capillary network nearby as well, with the exception of plants because they don't have blood. So some of the key terms linked to the gas exchange and ventilation topic that you need to be familiar with are breathing. So this is the movement of air in and out of the lungs. This is different to respiration because respiration is a chemical reaction that results in the release of energy in the form of ATP. Ventilation is the scientific term for breathing, so we'll be using the term ventilate quite a lot in this video. And gas exchange is the diffusion of typically oxygen and carbon dioxide in and out of cells. Now, it's not just the alveoli, that is just for mammals. So those are the key terms just to be familiar with, and these are the key structures that you need to know for the human gas exchange system. So the alveoli, bronchioles, bronchi, trachea and lungs, which are all labelled here. So if you need to pause just to note that down, then pause that just here. So we have a look at how humans ventilate. It involves the muscle, which is the diaphragm. It also involves antagonistic muscles, which are surrounding the ribs. And this is where we have the external intercostal muscles, but we also have internal intercostal muscles. Now, antagonistic means that as one muscle is contracting, the other relaxes. And we'll see that in our intercostal muscles, how the internal and external will always be doing the opposite. And that is what controls the movement of the rib cage in and out to help with ventilation. So a bit more about these internal and external intercostal muscles. Antagonistic muscles always occur as pairs, and the external intercostal muscle on the outside layer of the ribs, and they will contract and cause the rib cage to move out and air to flow in, or inspiration. The internal intercostal muscles, you can kind of see as it moves around 3D, they'd be on the inside layer around the ribs. And when these ones contract, that will pull the rib cage closer inwards, and therefore the air will flow out, or you'll be expiring. But just to have a summary then of what is happening to all of the muscles that are controlling the movement of the rib cage, and therefore whether you're inhaling or exhaling, or inspiring or expiring. So when you are breathing in, that is when your external intercostal muscles will be contracting, and that pulls the rib cage up and outwards. That means that the internal intercostal muscles are doing the opposite, they're relaxing. The diaphragm muscle contracts, and when that contracts, it causes it to move downwards and it becomes more flat. Now, as a result, that means that the volume of the thorax will be larger. So the volume increases. And by that, we don't mean the volume of gases. We mean the volume as in the cavity space. So the available volume, the actual space available. And because the lung volume, the thorax space is larger, that causes the pressure to drop, and because you now have a lower pressure within the lungs compared to outside, air is going to flow in. So that is what happens when you breathe in. Now when you exhale, it's exactly the opposite. So this time the external intercostal muscles relax, the internal will contract, and that pulls the rib cage down and back in. The diaphragm relaxes, which causes it to dome upwards. And as a result, the actual volume in the thorax, that space, is smaller. And because we have a smaller space or a smaller volume, that causes the pressure to increase. And that is why the air is forced out of the lungs. Now, the pulmonary ventilation calculation is one that you do need to know, and it's the total volume of air that's moved into the lungs during 1 minute. And the units would be decimeters cubed for volume and per minute. So to work this out, you would be doing the tidal volume times the ventilation rate. So tidal volume is the total volume of air, and the ventilation rate is how many times you breathe per minute. So then if we go on to the gas exchange and look at the alveolar epithelium. Once the gases are in the alveoli, so as a result of ventilation, this is where gas exchange occurs, and it's between the epithelium and the blood. Alveoli are tiny air sacs, and you have around 300 million in each lung. And that's actually why lung tissue floats compared to, let's say, muscular heart tissue, which would sink. So all of those alveoli air sacs make it float because it's so full of air. Now, the alveoli epithelium cells are also very, very thin, and that is to minimize the diffusion distance. So we can see here there's just one layer of cells and they're a thin layer. Each alveolus is surrounded by a network of capillaries, and this helps to maintain the concentration gradient, because as soon as that oxygen diffuses into the blood, it is then carried away in the capillaries and replaced by deoxygenated blood. Terrestrial insects, they do have a large surface area to volume because they're small. However, the issue they have to overcome is the fact that they have a waterproof exoskeleton. And that's an advantage to prevent water loss, but because it's made up of this hard fibrous material, it also means gases can't exchange across it. Now, insects don't have lungs, but they do have a tracheal system, which is where ventilation and gas exchange occurs. So their tracheal system, then, we have a look at how this differs to the human lungs. First of all, it includes a trachea, tracheoles and spiracles. The spiracles we can see here, they're the tiny holes, they're round valve-like openings that run along the abdomen, and this is where oxygen and carbon dioxide are going to be entering and leaving. So that is instead of like the nose or the mouth for humans. And those spiracles are going to be attached to trachea. So that's the next structure. And the trachea are tubes. They have rings within them to strengthen the tubes to stop them from collapsing, and that keeps the tubes permanently open. And then the trachea branch into smaller tubes, which are deeper in the abdomen of the insect, and those are called the tracheoles. And those extend throughout all of the tissue in the insect to deliver oxygen to all of the respiring cells. So here we have the spiracles connected to the trachea, and then the tracheal systems branching all the way through into the centre where the abdomen is and to all of the respiring tissues. Now, there's 3 methods of moving gases into that tracheal system. The first method is gases can just move in by diffusion, and that is because when the cells within the insect are respiring, they're using up oxygen, producing carbon dioxide, and that creates a concentration gradient from the tracheoles compared to the atmosphere. So some of the gases will simply diffuse in and out. The second method of gas exchange is mass transport, and this is where it's slightly similar to ventilation in humans, in that the abdominal muscles can contract and relax, and that is a way to ventilate, to move gases en masse in and out of the tracheal system. The final method is when the insects are in flight, the muscle cells will actually start to respire anaerobically. And when they do that, they produce lactate or lactic acid. And this lowers the water potential of the cells, and as a result, it draws water into the cells from the tracheoles. That decreases the volume in the tracheoles, and as a result, more air from the atmosphere is drawn in. So overall, the adaptations that terrestrial insects have for gas exchange are the fact that they have a large number of these fine tracheoles providing a large surface area. The walls of the tracheoles are very thin, and there's a short diffusion distance between the spiracles and the tracheoles, so that will speed up the rate of diffusion. And lastly, they use oxygen and they produce carbon dioxide, and that is how the steep concentration gradient is maintained. Now, the next challenge that the terrestrial insects have to overcome is limiting the amount of water that is lost at these gas exchange surfaces, because a gas exchange surface is also ideal for evaporation, and they do not want to be losing excess water. So here are the ways that water loss is prevented. Number one is the surface area where gas exchange actually happens is very small. So it's not the entire insect, we're just looking at the spiracle itself and then the tracheal system. That is a very small area compared to the whole volume of the insect, and therefore it's going to limit evaporation. The rest of the insect has this waterproof exoskeleton, and the spiracles can actually open and close, a bit like stomata on a plant, to reduce water loss also. So next is gas exchange in fish. Now, fish are waterproof and they have a small surface area to volume ratio. So those are the two reasons why fish have to have a gas exchange surface with adaptations. And for fish, it's gills. Now, fish obtain their oxygen from the water, but there is 30 times less oxygen dissolved in water than there is available in the atmosphere, in the air. So they have to have an additional adaptation to help maintain the diffusion and concentration gradient across their gills. So just as a reminder, for all of these gas exchange surfaces, they have to have these three features: a large surface area compared to the volume, a short diffusion distance, and they have to have a mechanism to maintain the concentration gradient. Now, those features can be used to work out the rate of diffusion using Fick's law. And this is just here. So diffusion is proportional to the surface area times the difference in concentration divided by the length of diffusion pathway. So you could get questions linked to this, testing that math skill. But let's have a look then at the fish gills to see how they have these three features maximised in their adaptations. So we're going to have a look at the fish gill anatomy. So there are 4 layers of gills on both sides of the head, and the gills are made up of stacks of gill filament. Each gill filament is covered in gill lamellae, and these are positioned at right angles to the filament. So this here is the lamellae, and this part is the filament. Now, because there are so many, that creates the large surface area. So that's the first adaptation ticked off. Because there's so many gill lamellae and gill filaments, it creates that large surface area. Now, when a fish opens its mouth while it is swimming, water rushes in and then actually comes out the side of their head, which means that the water rushes over the gills. So the water enters here and then it goes over the gills on both sides of the head, where they have these 4 layers of gills with the gill filaments and the gill lamellae. So the next thing is looking at the additional adaptations. We've talked about the large surface area to volume ratio, but the next thing is the fact that there is a short diffusion distance, and that is because of the network of capillaries in every gill lamellae. So we're zooming in here on the gill lamellae, and there is our network of capillaries within each one, and that provides a short diffusion distance. How the concentration gradient is maintained is by something called the counter-current flow mechanism. So the counter-current flow mechanism is where water flows over the gills in the opposite direction to which the blood is flowing through the capillaries. The reason this is an advantage is it makes sure that the equilibrium of the concentration of oxygen is never reached. And because we don't have equilibrium being reached, that means that oxygen is able to continue diffusing from the water into the capillaries in the gill lamellae across the entire gill lamellae. And that's in bold and underlined because that is the key marking point. Now that's demonstrated in these 2 diagrams here, where we look at concurrent flow in comparison to countercurrent flow. So in concurrent, not co-current, you'd have the water and the blood flowing in the same direction. So about halfway across the lamellae, you'd be reaching equilibrium, and therefore you wouldn't have any more diffusion of gases. Whereas here we have the water and the blood flowing in opposite directions, which means you should never actually have equilibrium, and there will always be a higher concentration of oxygen in the water compared to the blood, and that is why we maintain the concentration or the diffusion gradient across the entire gill lamellae. So lastly is looking at gas exchange in leaves. So you could be asked to look at some of the structures in the leaf. So we have the tissue layer palisade mesophyll, which is where photosynthesis mainly happens. And then we have the spongy mesophyll, where there's lots of air spaces. And we have the stomata, which is where the gases actually diffuse in and out of. So oxygen will actually diffuse out of the stomata if it's not being used in respiration. And carbon dioxide diffuses in because it's needed for photosynthesis. To reduce water loss by evaporation, stomata close at night when it's dark, and they'll open in the daytime when it's bright. And this is linked to the idea of photosynthesis because it's a light-dependent reaction. Now there are compromises though, and in particular linked to xerophytic plants, which are plants that are adapted to survive in environments with very limited water. So they have adaptations to minimise water loss because there's such little water available already. And over here, looking at this marram grass under the microscope, it demonstrates some of those adaptations really well. So the leaves aren't actually flat, they roll up, and we have stomata which are really deep and sunken in, and we have lots of tiny hairs sticking out. Now, all of those adaptations are to reduce water loss, because any water that is evaporating out is going to get trapped on these hairs, and the fact it's curled in, it gets trapped in this space. So it makes this area very, very humid, and if it's very humid, that will reduce further evaporation. Now, in addition to that, they also have a thicker cuticle to reduce water loss, and we can't see it on this particular image, but they do have a longer root network to be able to reach further distances to try and find any available water. So we now move on to digestion and absorption. And during digestion, large biological molecules are hydrolyzed into smaller soluble molecules which can be absorbed across the cell membranes. Now there's 3 biological molecules that you need to know the digestion of, and that's carbohydrates, lipids and proteins. So with the carbohydrates, there's actually more than one enzyme that's needed to hydrolyze them from the polysaccharides to the monosaccharides. We have amylases and membrane-bound disaccharidases. So digestion begins in the mouth, it will then continue in the duodenum, and then it's completed in the ileum. And the first thing is the amylase that is produced by the salivary glands is going to be doing the digestion in the mouth. But you also have amylase produced in the pancreas, and that is going to be releasing the amylase into the duodenum. And this hydrolyzes the polysaccharides into the disaccharide maltose, and that is by breaking the glycosidic bonds between the molecules in the polysaccharide. Then we have sucrase, and it could be lactase as well, which are membrane-bound enzymes that hydrolyze sucrose and lactose into monosaccharides. You could also have maltase, which would be hydrolyzing maltose into glucose. For proteins, these are large biological molecules, they're polymers, and they are hydrolyzed by 3 enzymes. We have the endopeptidases, which hydrolyze the peptide bonds between amino acids in the middle of a polymer chain. We have the exopeptidases, which are going to do hydrolysis and break the peptide bonds between amino acids at the end of the chains. And then the membrane-bound dipeptidases will be breaking the peptide bonds between 2 amino acids. So it hydrolyzes those dipeptides. Now, where this is actually happening is firstly in the stomach. So that's where it begins. It continues in the duodenum, and then it's fully digested within the ileum. Lipids are digested by lipase or lipase, and also through the action of bile salts. Now, lipase is produced in the pancreas, and it can hydrolyze the ester bonds in triglycerides to form monoglycerides and fatty acids. What the bile salts do after they've been produced by the liver is emulsify lipids to form tiny droplets called micelles. and these increase the surface area available for the lipase to bind onto and therefore act on. So they're going to be making this digestion more efficient. So we can see here the physical and the chemical digestion. Physical is the emulsification and that micelle formation. Chemical digestion is the action of the lipase. So the physical, this is where the lipids are coated in bile salts to create an emulsion. Many of those small droplets of lipids provide a larger surface area to make hydrolysis faster. And as we said, the chemical is just the action of lipase. Now, a micelle is a vesicle formed of fatty acids, glycerol, monoglycerides and bile salts, but we're going to have a look at how that aids in the absorption of lipids. So lipids are digested into monoglycerides and fatty acids by the action of lipase and the bile salts. And that is how we create these tiny structures, which are the micelles. When the micelles encounter the ileum epithelial cells, due to that non-polar nature of the fatty acids and monoglycerides, they can simply diffuse across the cell surface membrane to enter the cells of the epithelium. Once in the cell, these will then be modified back into triglycerides inside of the endoplasmic reticulum and the Golgi body. And then they can form vesicles and be released from the cell into the lacteal and be transported around the body. So absorption in mammals is taking place in the ileum. And this is where it links to the villi and the microvilli and the adaptations that these structures have. So the ileum wall is covered in villi, and they have thin walls surrounded by a network of capillaries. And they also are covered in epithelial cells that have even smaller microvilli on them. And that is creating that large surface area. The fact that the walls are so thin is providing a short diffusion distance, and that network of capillaries is maintaining the concentration gradients. Now this actually links to as well topic 2 when you learnt about co-transport. And that is because the monosaccharides like glucose and amino acids can only be absorbed by active transport in the form of co-transport. And that is because there's usually a higher concentration of either amino acids or glucose already in the epithelial cell. So in order to be able to move even more from the ileum into the epithelial cell, we require this co-transport. But that theory is covered in topic 2. Now, hemoglobin is involved in the mass transport of oxygen around the body. And it's an example of a quaternary structure protein because it's made up of 4 polypeptide chains. You also have a range of different types of hemoglobins. One that we'll be looking at is myoglobin, which is found in the muscle tissue in vertebrates and also in fetuses. The oxyhemoglobin dissociation curve is a way to look at how hemoglobin behaves in different conditions. Oxygen is loaded in regions with a high partial pressure of oxygen. So what that means is when hemoglobin is in areas with a lot of oxygen available, such as the alveoli, it will be able to pick up lots of oxygen. In regions with a low partial pressure of oxygen, for example respiring tissues, hemoglobin unloads the oxygen. And that is how we get the shape of this oxyhemoglobin dissociation curve. We can see at the different partial pressures how saturated hemoglobin is with oxygen. And the more saturated it is, that means it must have loaded up with more oxygen. The less saturated it is, it must have unloaded oxygen, and that's why it's not holding very much anymore. So we can see here we've got our respiring tissues compared to the alveoli. Now this graph also demonstrates cooperative binding. And this cooperative nature of oxygen binding to hemoglobin is due to the hemoglobin changing shape when the first oxygen binds. It then makes it easier for the subsequent oxygens to bind. Now you can also see dissociation curves to demonstrate the Bohr effect. And the Bohr effect is when a high carbon dioxide concentration or partial pressure causes the oxyhemoglobin curve to shift to the right. The affinity for oxygen decreases, and that is because the acidic carbon dioxide changes the shape of the hemoglobin slightly, and therefore it means the hemoglobin behaves differently and it's more likely to unload the hemoglobin even at the same partial pressures. So we can see here we've got three curves at different pHs, and the lower the pH, the more carbon dioxide there would be present. So we can see here at pH 7.6 compared to, let's say, 7.2, the curve has shifted to the right, and even at the same partial pressure, so we'll pick 20, we can see that the saturation is only about 25%, but for this one it's just under 60%, and that is because of this Bohr effect. So low partial pressure of carbon dioxide would typically be in the alveoli because you are exhaling that carbon dioxide. A high partial pressure of carbon dioxide would be at respiring tissues because carbon dioxide is produced in respiration. Now that would be an advantage because it means that hemoglobin will behave differently, it will unload oxygen more readily, and therefore it's unloading the oxygen at the respiring tissues. Now, different animals have hemoglobin adapted to their particular needs and environments also. And that is one thing that you could get application questions on. So a fetus, and this is a human fetus, they will have myoglobin or fetal hemoglobin, and the fetal hemoglobin has an even higher affinity for oxygen even at the same partial pressures compared to adult hemoglobin. And that's an advantage because it means that as the blood is circulating through the umbilical cord, the fetus's hemoglobin is able to load the oxygen off of the mother's adult hemoglobin. Llamas are found at high altitudes where we have very, very low partial pressures of oxygen. So for the llama, we can see that they also have hemoglobin that has a higher affinity for oxygen, even at lower partial pressures. So we can see that would mean that if there isn't very much oxygen available, which there wouldn't be at a high altitude, then the hemoglobin is still able to load up. Animals like doves, for example, their needs are that they need more oxygen to match their faster metabolism because they're flying so much, they need oxygen for the muscle contraction. So the hemoglobin of a dove, the curve actually is shifted to the right compared to human hemoglobin. And that means that the hemoglobin has a lower affinity for oxygen, and therefore it will more readily unload the oxygen which is needed for respiration. Earthworms are a different example, so they will be underground a lot where there's very low partial pressures of oxygen. So they have hemoglobin that has very high affinities even at low partial pressures, so that their hemoglobin can load up with whatever oxygen is available. In mammals, the circulatory system is described as closed and double. Closed meaning that the blood remains within the blood vessels the entire time, and double referring to the fact that the blood passes through the heart twice in each circuit. So there is one circuit that delivers blood from the heart to the lungs, and the other circuit delivers the blood from the heart to the rest of the body. Mammals are required to have a double circulatory system to manage the pressure of blood flow. The blood flows through the lungs at a lower pressure, and this is to prevent damage to the capillaries in the alveoli, but it also means that the blood will flow at a slower speed, so there is more time for gas exchange. The oxygenated blood from the lungs then goes back to the heart and it's pumped out at a higher pressure to the rest of the body. And this is important to make sure that the blood is able to reach all of those respiring cells in the body. Now, the key blood vessels that you need to know about are, first of all, the coronary arteries, and these are the arteries that cover the heart itself to supply the heart muscle or cardiac muscle with oxygenated blood. You also need to know the 4 blood vessels that are delivering blood into and out of the heart: the vena cava, aorta, pulmonary artery, and the pulmonary vein. You need to know the blood vessels that deliver blood to the lungs and carry it away, so the pulmonary artery and the pulmonary vein. So any time that you see pulmonary, that is referring to the lungs. And you can see here that the pulmonary artery is carrying blood away from the heart to the lungs. The lungs will then oxygenate the blood, and the pulmonary vein is delivering that blood back into the heart. The kidneys, we have the renal artery and the renal vein. So where you see the word renal, that is referring to a blood vessel attached to the kidneys. Those major blood vessels are connected within this double circulatory system via arteries, arterioles, capillaries and veins. The cardiac muscle has a range of special features. Now the walls of the heart have a very, very thick muscular layer so that it can contract with high force to deliver high pressure blood to all of the body cells. Now the unique properties that the cardiac muscle has is, first of all, it's myogenic, and that means it can contract and relax without nervous or hormonal stimulation. It also never fatigues, so as long as it has a constant supply of oxygen and glucose, it will be able to respire aerobically. The coronary arteries we can see here, they are what supply the cardiac muscle with this oxygen and glucose so that it never fatigues. They branch off from the aorta, which we can see here. And if one of those coronary arteries was to become blocked, that would then mean that the heart muscle or the cardiac muscle wouldn't be receiving the oxygen or glucose. Therefore, the cardiac muscle wouldn't be able to respire and it would stop contracting. And that would cause a myocardial infarction, or in other words, a heart attack. So you need to know some of the key structures of the heart. First of all, there are 4 chambers. We have 2 atria at the top and we have the 2 ventricles at the bottom. The atria have thinner muscular walls, and that's because they don't need to contract with as much force because they're only delivering the blood from the atria into the ventricles. They also have elastic walls so that they can stretch when the blood is entering. The ventricles have much thicker muscular walls, and that is so they can contract with more force and pump the blood out at higher pressure because they are carrying the blood further distances, either to the lungs or to the rest of the body. Now the right ventricle is pumping the blood to the lungs, and that is at a lower pressure, as we said, to prevent damage to the capillaries and to go at a slower speed. So comparatively, the right ventricle wall has a thinner muscular layer. The left ventricle has a much thicker muscular layer in its wall because it has to contract with more force to pump the blood at high pressure around the body. Highlighted here we have the 4 key blood vessels. We've got the aorta and the pulmonary arteries, the vena cava and the pulmonary veins. But for some of them there's actually multiple. So we can see we have the superior vena cava and the inferior vena cava. We also have the pulmonary artery, which is carrying the blood away from the heart to the lungs, but we have one coming out of the left and one out of the right. Same with the pulmonary veins, which is carrying blood into the left atrium from the lungs. We have one on the left side, one on the right side. And the reason for both of those is we have a right lung and a left lung. The aorta is carrying blood from the left ventricle to the rest of the body. Now, some of the ways just to try and remember what each of these are doing are, first of all, if you see veins, veins are carrying blood into the heart. And vena cava means vein, and cava means body. So that is carrying the deoxygenated blood from the body into the right atrium. Pulmonary, we said, means lungs, and again it's a vein. So it's carrying oxygenated blood from the lungs into the left atrium. Arteries, think A away, it's carrying blood away from the heart. So pulmonary artery is carrying deoxygenated blood from the right ventricle to the lungs. And the aorta is the major artery, which is carrying oxygenated blood from the left ventricle to the rest of the body. Now the valves that you need to know about are the semilunar valves, which are in the aorta and the pulmonary artery, as well as the atrioventricular valves, which are between the atria and the ventricles. Sometimes you'll see those called the bicuspid and the tricuspid valves. And we can see these labelled here on the diagram of the heart. Now valves are to prevent the backflow of blood, and they do this by only opening when the pressure is higher behind the valve compared to in front. So if the pressure is higher in front, it causes the valve to shut, and that is how it stops the blood from flowing backwards. The septum is running through the middle of the heart to separate the blood on the deoxygenated and the oxygenated side, and that's to help to maintain a high concentration of oxygen in the oxygenated blood to make sure that these diffusion gradients are maintained so that diffusion can occur at respiring cells. Looking at the blood vessels that are connecting all of the major blood vessels together, we have the arteries, arterioles, capillaries and veins. Arteries, as we said, think A away, they carry blood away from the heart towards the arterioles. The arterioles are smaller than arteries and connect to the capillaries. The capillaries connect the arterioles to the veins, and the veins will then carry blood back into the heart. So we can see here our arteries connected to our arterioles. We then have this capillary bed or network of capillaries connecting to the venules and then the vein. So the structures of the arteries and vein are quite different because of the fact that one is carrying blood away from the heart and one's carrying it into, so the pressure of the blood they're carrying will vary. So arteries have a much thicker muscular layer compared to the veins, and that is so that constriction and dilation can occur to control the volume of blood flowing through them. They also have a thicker elastic layer than the veins, and that's to help maintain the blood pressure. So the walls can actually stretch and recoil in response to the heartbeat. So overall, the thickness of the wall is thicker because of those two thick layers, but also it stops the blood vessels from bursting at that high pressure. So we can see here how thick those walls are. They also don't have any valves. So in comparison, veins have a much thinner muscular layer, thinner elastic layer and thinner walls in general, and that is because the blood is at lower pressure, so they're not at risk of bursting. However, because the blood is at lower pressure, they do require valves to help prevent the backflow of the blood, to make sure that the blood is going to be pumped back into the heart. The capillaries are very, very narrow in diameter, and this is to make sure that the blood speed is going to slow right down as it's passing through the capillaries. And that is to allow time for gas exchange and tissue fluid formation in the capillaries. So we've already talked about the arteries and the veins, but if we now add in the arterioles and capillaries, the arterioles are thicker than in the arteries, the muscular layer, and that is to help restrict the blood flow to the capillaries. The elastic layer is thinner than the arteries, and that is because the pressure is now slightly lower. And overall, the wall is thinner compared to the arteries because the pressure is slightly lower, but they still don't have valves. Capillaries do not have any muscular or elastic tissue layer. They are only one cell thick, and that is to make sure there's a really short diffusion distance, because the function of the capillaries is that is where exchange of materials between the blood and the cells occur. The cardiac cycle is split into 3 stages. Now people pronounce this differently, so I'm going to say both: diastole or distole, atrial systole or atrial systole, and ventricular systole or systole. And we're going to go through what happens at each stage. So in diastole, the atria and ventricle muscles are relaxed. And this is when blood will enter the atria through the vena cava and the pulmonary vein. And the blood flowing into the atria then causes an increase in the pressure because we've now got a larger volume of liquid there. Then we get atrial systole occurring. And that is when the atria muscles contract, and that increases the pressure even more in the atria. And because we now have this high pressure, that causes the atrioventricular valves to open, and blood moves from the atria into the ventricles. And at this stage, the ventricle muscles are relaxed. We have ventricular diastole. The last stage then we have is ventricular systole, which is when the ventricle muscles contract. And that happens after a short delay, and it will increase the pressure beyond that of the atria, which causes these atrioventricular valves to shut. But when they've contracted high enough to increase the pressure above that of the atria and pulmonary artery, the semilunar valves will open, and that causes blood to be pumped out of those two blood vessels. You could be asked to calculate the cardiac output, and that is the volume of blood which leaves one ventricle in 1 minute, and it can be calculated by doing the heart rate times the stroke volume. And the heart rate is beats of the heart in 1 minute. Stroke volume is the volume of blood that leaves the heart each beat, and that is in decimeters cubed. Tissue fluid is a fluid that contains water, glucose, amino acids, fatty acids, ions and oxygen, and it's the liquid that is forced out of the capillaries to bathe the cells, to make sure the cells are getting all of the minerals, nutrients that they require from the blood. Now it's formed due to the fact that capillaries have very, very small gaps between each of the cells that make up the capillary walls. And as the blood enters the capillaries from the arterioles, the smaller diameter results in a very, very high hydrostatic pressure of the blood. And because we have this high hydrostatic pressure and tiny gaps between the cells that make up the capillary walls, water and small molecules are forced out. So glucose, amino acids, fatty acids, ions, oxygen are all forced out of the capillary to the surrounding cells. And that is called ultrafiltration. So the molecules that can be forced out, we just went through all of those, but anything that is too big cannot get out of those tiny gaps between the capillary cells, and that would be things like the red blood cells, platelets and large proteins. So those will remain in the blood. And the fact that they remain in the blood is actually what enables the liquid from the tissue fluid to be reabsorbed again. So towards the venial end of the capillary, which is over here, the hydrostatic pressure has decreased, and that's because so much liquid has been forced out. The pressure within the capillary has now dropped by the time we've got to the venial end of the capillary. However, we don't have as much liquid, but we have lots of those large proteins that have remained behind, which has lowered the water potential of the blood in the capillary compared to the water potential in the liquid surrounding the cells, which is the tissue fluid. And as a result, that liquid is going to, or the water is going to re-enter by osmosis, and it will carry with it waste that has dissolved in that tissue fluid that was released from these cells. So things like carbon dioxide and urea. So that gets absorbed into the capillaries by osmosis, and then it'll be transported around the body to be removed as waste. Now, not all of the liquid will be reabsorbed by osmosis, because eventually equilibrium will be reached and the water potential inside of the capillary and of the tissue fluid will be the same. So any of that water that doesn't get reabsorbed by osmosis will enter the lymphatic system instead, and then when we have it transported around the lymphatic system, it will eventually get to one of the lymph vessels near the heart where that liquid is drained into the blood again, and that is how we don't run out of liquid in the blood. So the last bit of topic 3 is mass transport in plants, and we start with the mass transport of water, looking at first of all transpiration. So transpiration is the loss of water vapour from the stomata. So water evaporates out of stomata on the leaves. And there are 4 key factors that affect the rate of transpiration. First of all, light intensity. The more light there is present, the more stomata will open, and therefore there's a larger surface area for evaporation. If it's hotter, that means that the water molecules will have more kinetic energy, and therefore they'll be moving faster and evaporate at a faster rate. With humidity, the more water vapour that is in the air, that will actually make the water potential more positive outside of the leaf, and it reduces the water potential gradient. So what that means is the more humid it is, the less transpiration there will be occurring. With wind, because the wind is carrying away air that contains water vapour, the wind will actually be maintaining the water potential gradient. So the more windy it is, the faster the rate of transpiration. So how water actually moves from the roots all the way up the xylem to transpire out of the stomata links to this idea of the cohesion tension theory. So water has to move up the plant from the roots against gravity, and that can be a very, very large distance if it's a huge tree. And this is possible because of the cohesion, the capillarity or adhesion, and root pressure. So if we look at cohesion first, this links to the theory of water. Water is dipolar, meaning we've got two slight charges, two different charges, a slight negative on the oxygen and a slight positive on the hydrogen atoms. And that means hydrogen bonds can form between the hydrogen and oxygen of different water molecules. And it creates this cohesion or sticking together of the water molecules. Now that's an advantage because as the water moves up the xylem, that means it's all bonded together with these hydrogen bonds, and it moves up as a continuous column of water. And it's much easier to pull up a column than it is individual water molecules. The next idea was this idea of adhesion, whereby the water molecules will actually also stick to the walls of the xylem. And if you have a really narrow xylem, that will increase this capillarity effect, and therefore the liquid will be moving up more just from this sticking, this adhesion effect. So the narrower the xylem, the easier it's going to be to transport the liquid up the xylem against gravity. Root pressure is the final concept, and this is as the water moves into the roots by osmosis, it increases the volume of liquid inside the roots, and therefore the pressure inside the root increases as well. And we call that root pressure. And that creates this positive pressure, which means it's a pushing force. Because we have lots of water in the roots, it pushes all of the water above it upwards, so it helps push the water up the xylem. So those three concepts all work together for the cohesion tension theory, and that's how water moves up against gravity. So just have a look at this as a whole. The first thing that happens is the water evaporates out of the stomata, and as that water vapour is leaving, that is then leaving behind this lower pressure because liquid has been lost. And that creates this negative pressure, or in other words, a pulling force. And that pulls the water column up the xylem. And because of cohesion, we have that water column whereby all the water molecules are stuck together. The water molecules are also adhering or sticking to the walls of the xylem to help pull the column upwards. And as the column of water is pulling up, it also creates tension in the xylem, which actually pulls the xylem inwards, making it narrower, which increases that impact of the capillarity or the adhesion, and it helps to move the water up even more. Now, the second type of mass transport in plants is looking at how the organic molecules like glucose produced in photosynthesis are transported around. And this now happens in the phloem. Now, the phloem tissue contains two key cells. We have the sieve tube elements and the companion cells. And the sieve tube elements, which are here, are living cells, but they don't contain any nuclei, and they're very few organelles. And that is so it's pretty much hollow to make it easy for the solutions to transport through the tube. The companion cells are on the outside, and they provide the ATP required for active transport of the organic substances. So they have all the organelles that the sieve tube elements don't, so they can provide the resources needed. Now, the transport of these organic molecules within solution is often explained using this source to sink model. So we have here the xylem next to the phloem, and the source in this model would be a photosynthesizing cell. The sink, which is where the sugars are going to be delivered, is a respiring cell. So at the source, we have photosynthesizing cells, and the sucrose or glucose that would be made in photosynthesis is going to lower the water potential of those cells. and therefore any surrounding water in the plant or from the xylem is going to be entering those cells by osmosis. At the other end, where we have our respiring sink cells, because they'll be using up those sugars in respiration, there will be a more positive water potential inside of the cell compared to outside, and therefore water is going to leave the respiring cells by osmosis to other cells in the plant or even the xylem. So the effect that has is there'll be an increase in the hydrostatic pressure in the source cell, but a decrease in the sink cell. And because of those pressure changes, the liquids that are within that source cell will be forced by that high hydrostatic pressure through the xylem all the way to the sink cell. Now that is part of the story, but also you need to know the translocation steps. So how those sugars within the leaf cell actually make it into the phloem for that pressure then to actually move the liquid along. So photosynthesis is occurring in the chloroplasts, in the leaves, and we're calling those the source cells. That, we said, creates a high concentration of sucrose. Now, as well as that affecting the water potential, that also means that the sucrose can diffuse down its concentration gradient into the companion cell by facilitated diffusion. We then get active transport of protons or hydrogen ions from the companion cell into the space within the cell walls, and that uses energy because it's active transport. Now that creates a concentration gradient, and therefore the protons move down their gradient via carrier proteins into the sieve tube elements. And the co-transport of sucrose with those hydrogen ions occurs via a protein co-transporter, and that is how the sucrose goes from being in the companion cell into the phloem, even though there is a high concentration often of sugar or sucrose in the phloem already. So the next step then is we're looking at the movement of that sucrose within the phloem, sieve tube element. So the increase of sucrose in the sieve tube element is going to lower that water potential, and therefore water enters those sieve tube elements from the xylem vessels, which are tightly compacted next to the phloem. The increase in water volume in the sieve tube element at this position will increase the hydrostatic pressure, and this is where we have the idea of source to sink. It causes the liquid to be forced from the source area down to the sink cells. Lastly, the sucrose is then used in respiration at the sink, or it might be stored away as starch if it's not currently required. But that means more sucrose is actively transported into the sink cells, and that is going to cause the water potential to decrease. And as a result, we'll have water moving by osmosis from the sieve tube elements into the sink cell. Some water will also be returning from the sieve tube element into the xylem. The removal of that water is decreasing the volume in the sieve tube element, and therefore the hydrostatic pressure decreases. So the movement of soluble organic substances is due to the difference in the hydrostatic pressure between the source and the sink end of the sieve tube element. Now you could be asked about two particular investigations that prove translocation. The first one is called traces, and this is where we have tracing involving radioactively labelling carbon. Plants are provided with only radioactively labelled carbon dioxide, and over time they'll be absorbing that in through the stomata, using it in photosynthesis, and the organic substances like the sugars created will all contain that radioactively labelled carbon. Thin slices from the stems are then cut and placed on X-ray film that will turn black when exposed to radioactive material. When the stems are placed on the X-ray film, the section of the stem containing the sugars turn black, and this highlights where the phloem are, and it can show the sugars are transported in the phloem, and it also means you can track the route that is taken. Ringing experiments is when a ring of bark and phloem are peeled and removed off the trunk, like we can see here. The result of removing the phloem is that the trunk swells above the removed section, and analysis of the liquid in this swelling shows it contains sugar. So this shows that when the phloem is removed, the sugars cannot be transported, and it therefore proves the phloem transports sugars. So that is it for topic 3. I hope you found it helpful. If you did, please give this video a thumbs up. And if you want to learn any of those in more detail, then click these links to see the full playlist.